This is a video about how to draw the phospholipid bilayer for IB Bio. So we start off with the structure that makes up the bilayer. This is one phospholipid. It is phospho because the top bit is phosphate. Phosphate is PO4 3-, so it has a negative charge. There is a negative charge at the top, so we can think of it as being negative at the top. It's a lipid because it has 2 fatty acid tails, which are carbon and hydrogen in a long tail coming off. They may have some double bonds which would get rid of this, but they are just carbon and hydrogen. Oh, let's get rid of that as well. So because it's a lipid tail, the lipid tail, this section is non-polar. It doesn't have charges. Whereas the phosphate head, because it's a negative charge, the phosphate head is polar. Non-polar we can also call hydrophobic, whereas the polar we could call it hydrophilic. If we think about these structures with a head which is hydrophilic, water loving, this part will be able to attract to a water molecule, and it would be actually hydrogen bonding to the hydrogen part. So let's draw that the right way around. Hydrogen, hydrogen, the oxygen would attract. Now, if we have water, the water is outside cells and inside. There is floating around lots of water molecules. And then we have the membrane, and inside, in the cytoplasm, is also lots of water. So there is water outside and there is water inside. Now if we think about how these must arrange then, if this part likes water, we will get the heads arranging facing the water, which means the tails will point inwards, because they are non-polar. So this, the fatty acids, arrange in the middle of the membrane. The polar heads are facing water, and that's outside and inside. So let's draw a more detailed structure of the membrane. So we would draw ourselves some heads. We would also draw some this side. They can have a gap in the middle. We don't have to have them all touching. We don't really have to have the heads touching. They could. So we've got, if we label this, we have a glycerol and phosphate head. we have fatty acid tails. And there are 2 fatty acid tails that we could label there. We can also label this as being hydrophobic, non-polar. And this is hydrophilic, polar. The fatty acid tails are in the middle. The hydrophilic, phosphate and glycerol are facing outwards towards water. The whole thing, one of these, is a phospholipid. And we can see that we've got 1 layer and 2 layers. So we can label this as a lipid, phospholipid bilayer. Okay, so I've labelled these things here. Let's imagine that there's some more continuing here. And let's draw some other structures in the membrane. So the membrane has a big section all through here. This is all non-polar, which means we have a large amount which is non-polar. And that means that if a molecule is polar, and especially if it is large and polar, then it can't get through. It's going to be blocked here. It won't be able to push through. So something that's large and polar would be glucose. Glucose has C6H12O6. With the 6 oxygens, it has a lot of polarity. It is polar. And that means it can't move through. So we're going to draw a way for our glucose to get through the membrane. What glucose is going to do is use a channel protein. I'm going to draw it. Now, if we were looking at it from above, it would be more like something with a hole down the middle. So that would be the hole where glucose is going to move down. This channel protein is going to cross the membrane. If it crosses the membrane, we call it an integral. It crosses the membrane. So we've got an integral protein. We also call it, because it has a channel down the middle, it is a channel protein. So one of the things that we'll have is channel proteins for things like glucose to go down through the middle. We might also have as well proteins where they are stuck to the surface, one surface or another surface. So let's draw one down here. We're going to draw one here which is attracted to the heads. This is a extrinsic protein. It's outside. We could also call it a peripheral protein. So we're going to draw one of those. We also get things in the membrane which could be sticking through. I'm going to draw it all the way through. And then up out of them, I'm going to draw some little hexagons to be sugars. Up out of the membrane, we have got these sugars sticking up, and I'll draw some going one way and one the other way. We've got sugars sticking up, And because it's a sugar, we use glyc. Glyc is the prefix. It's a glycoprotein. This glycoprotein is sticking up out of the membrane. It may be used as a receptor. So it could be a receptor protein, a glycoprotein sticking up out of the membrane. Okay, so we've labelled a number of things. We've got our channel protein, we've got an extrinsic protein here that's on one side of the membrane, or a peripheral protein. We have got another, it's integral because it crosses the membrane, but now it's got a glycoprotein on. The final thing that we want to label is that we have another thing which sits in the membrane next to the hydrophobic tails. And this final thing to label, I'll label it up here, is cholesterol. So cholesterol is something that sits amongst the tails, and it gets in the way of the tails moving, so it has a role in regulating fluidity. Okay, so that, I've added a lot of detail to it. We've got our phospholipid bilayer made up of phosphate and glycerol head that's hydrophilic, that points towards the water that is outside the cell and points towards the water that's inside the cell. We've also got our lipid bilayer, because it's two layers of these phospholipids, with the fatty acid tails, which are non-polar, or we could say hydrophobic. Hydrophobic in the middle. We've talked about a channel protein that's integral, that allows large and polar things like glucose. It might also be a channel protein for small polar things like sodium ions or potassium ions to go through the membrane. We have got a glycoprotein, which could be acting as a receptor, and an extrinsic or peripheral, which is on one side of the membrane. So that is how to draw the phospholipid bilayer and all the features of it.